All right, first up today we have Eva. She was born and raised in Puerto Rico. She received a Bachelor of Science degree in Industrial Biotechnology from the University of Puerto Rico, Mayaquez, and a Doctor of Veterinary Medicine degree from Cornell University. She is currently the 2019 and 2020 Aquatic Animal Health Fellow at the University of California, Davis, and is currently working on her last rotation of the fellowship at Hub's SeaWorld Research Institute. During the first portion of her fellowship, she conducted a research project focused on pet fish medicine under the guidance of Dr. Esteban Soto at the Aquatic Animal Health Lab at UC Davis. And her presentation today is the fruit of that labor. She had so much fun doing this that she is going to be returning to Davis to start a PhD in microbiology this upcoming fall semester, focused on infectious diseases in fish. So you can take it away, Eva.
Thank you. Hello, everyone. My name is Eva Marie Quijano Cardé, and like Nick mentioned, I'm the current Aquatic Animal Health Fellow at the University of California, Davis. During the first 6 months of the fellowship, I had the opportunity to conduct a research project under the guidance of Dr. Esteban Soto, and today's presentation is a summary of those findings. So get ready for the pharmacokinetic and efficacy study of acyclovir against cyprianid herpes virus 3. Let's first talk a little bit about koi. I know most of you, if not all, know about these awesome little guys, but let's discuss shortly their importance. Koi are the ornamental variety of the common carp, and just like dogs, they come in many different varieties or breeds, if you will. They are very responsive to human interaction, which makes them really amazing pets. In fact, koi are one of the most common ornamental pet fish in California and are definitely the most frequently assessed pet fish here at UC Davis. And just as perspective, koi and goldfish are actually the most popular freshwater ornamental fish worldwide. With an increase in studies regarding fish's ability to help decrease stress in people and also help Alzheimer's patients maintain focus for longer periods of time, it's expected that we as veterinarians will continue to see more fish patients, and therefore, as scientists, should be paying more attention to health problems that require more research. And it's precisely that kind of thinking that brought us here. If you attended the very first seminar in this series, you learned about this infectious agent from Dr. Tolo, but we will go over it briefly. Around the year 2000, a herpes virus, later named cyprianid herpes virus 3, more commonly known as koi herpes virus, or KHV, was identified as the causative agent of several mass mortalities of carp and koi throughout the second half of the 1990s. This virus is now notifiable to the World Organization of Animal Health. KHV most commonly causes lethargy, inappetence, increased mucus secretion, and dyspnea secondary to gill necrosis as early as 3 days post-infection. Hyperemia, gill and skin discoloration, and ophthalmia and neurologic deficits are not uncommon on later stages of the disease. And when all is said and done, a KHV can cause between 80 and 100% mortality. Similar to other cyprianid viruses, KHV is a temperature-dependent virus that only causes disease at the permissive temperatures of 15 to 28 degrees Celsius, which is why temperature modification has been used therapeutically in the past. Unfortunately, due to the latency capabilities of herpes viruses, these exposed carriers are still vulnerable to subsequent outbreaks and can infect naive fish introduced into the system. In addition, temperature modifications can be very stressful for sick fish and may not be possible in some systems. While an attenuated live vaccine and an inactivated vaccine have been developed and are used in some countries, the risk of reactivation and low protection have hampered their use in the US, and there are no approved or recommended effective feasible treatments to reduce mortality. There have been 2 important studies where antivirals were investigated. In 2017, a team in Thailand investigated a plant extract called Clinicanthus nutans. In their in vivo experiment, they were able to demonstrate that mortality could be significantly decreased by administering this extract pre-infection or up to 24 hours post-infection. Unfortunately, this compound is not regulated and cannot be obtained in a reliable commercial formulation. Later in 2018, a team in Poland decided to try acyclovir against KHV in vitro, and their findings demonstrated that this antiviral is capable of decreasing cytopathic effects, viral load, and viral gene expression when added 2 hours post-infection. Considering that acyclovir is an approved antiviral by the FDA for the treatment of herpes simplex virus 1 and 2 in humans, it's commercially available and a good option for extra label use in pet fish under veterinary care. So let's talk about the drugs for a minute. The image on the right is a depiction of acyclovir's mode of action. This compound is a nucleoside analog that, once phosphorylated, will be added with more affinity by the viral DNA polymerase to the growing viral strand, effectively halting viral replication. Considering the supportive potential of acyclovir against KHV, it's only expected that we would pick this antiviral as a candidate. We also decided to try a second antiviral called cidofovir, which is a nucleoside analog as well. The advantage of cidofovir is that it does not require the initial phosphorylation step, which is made by a viral kinase. So when you have resistance against acyclovir due to mutations in the viral kinases, cidofovir could still be effective. In addition, cidofovir has secondary active metabolites in humans that allow for a longer effective time. So the objectives of the study were to test the cytotoxicity for both acyclovir and cidofovir on COI cells to determine if either antiviral had a significant efficacy against KHV in vivo, and to obtain plasma pharmacokinetics of the successful antiviral in COI after a single intrasomatic injection. With that in mind, let's dive in. For the cytotoxicity assay, COI fin cells, or KF1 cells, were grown using standard cell culture media on T75 flasks from stock until 100% confluency was obtained. Then they were plated on 96 well plates at approximately 90% confluency and allowed to attach for 24 hours. Antiviral solutions of acyclovir and cidofovir were made in sterile phosphate buffered solution, or PBS, and used within 1 hour to obtain triplicate wells of 5 micromolar to 1500 micromolar of each drug. Cells were then incubated with the antiviral for 24 hours. We use the Promega Cytotox 96 non-reactive cytotoxicity assay and an automated plate reader to measure lactate dehydrogenase. LDH is released when cell membranes are compromised or with cell lysis, so its release can be used as an indication of cytotoxicity. We included negative controls, which were treated with sterile PBS, and positive controls, which were lysed KF1 cells, to calculate cytotoxicity percent. And we did this experiment twice to have technical replicates. A nested one-way ANOVA and Dunet's multiple comparison analysis was used to determine statistical significance, which was considered at a p-value of 0.05 or less. And these are the results from this experiment. The solid dots represent the mean cytotoxicity percentage at each exposed concentration with standard error of the mean. The dotted line represents the mean cytotoxicity percentage of the negative control, while the solid lines represent its 95% confidence interval. The different experimental groups are color-coded just below the title. As you can appreciate, there was no statistically significant difference between the treated groups and the negative control, which suggests that both acyclovir and cidofovir at the tested concentrations are non-cytotoxic to KF1 cells. It's worth it to mention that most of these concentrations are much higher than what we would expect to obtain in vivo, considering the human data available. But even with these high numbers, we could not identify a median lethal dose, or LC50. For the challenge experiment, we cultured KHV by infecting KF1 cells grown as previously described in the cytotoxicity slide. We used a stocked strain from a previous outbreak here in California. We then used the crystal violet virus plaque assay to determine plaque forming units and calculate titration for exposure, which was 0.6 PFU per mil. We used 420 koi fingerlings in total for this challenge, averaging 8 grams in weight, and allowed them to acclimatize to holding for about 2 months while they gain a little bit of weight. First, we exposed 30 koi fingerlings to KHV via a 6-hour immersion. In addition, we exposed 30 koi fingerlings to the same volume of sterile culture media to be used in the negative control group. Then we monitored them for 9 days to give the virus a chance to increase its load, at which time we confirmed a 79% KHV infection prevalence via qPCR of fin clips. All sampled negative controls tested KHV negative. Then the fish were divided among the appropriate experimental groups for a cohabitation model. This type of infection model was selected because it would represent more accurately a natural outbreak. We monitored the groups closely until mortality started happening in the infected groups, which took approximately 2 weeks. And we wanted to wait until morbidity or mortalities occurred because this is when we would be called as veterinarians. At this time, we confirmed a 50% KHV infection prevalence after cohabitation via qPCR of fin clips. All sampled negative controls tested KHV negative, and all mortalities tested KHV positive. We then administered the appropriate treatment per group under buffered MS222 anesthesia, and each group had a replicate tank, had 6 replicate tanks with 15 fish each. The doses are on the screen and were based on intravenous dose recommendations in humans against herpes viruses. Just a note that we adjusted the drug concentration so that both groups of fish would receive a similar volume, even with different doses. The positive and negative control groups received a 10 ml per kg volume of sterile PBS solution, which was the diluent of the antivirals. I want to mention that the small weight of the koi fingerlings for this study is a limitation to have in mind, as well as for the PK study later, because it reduces the accuracy of the small volume we were injecting. Mortality was then recorded for 30 days and gills collected from mortalities for KHV confirmation. At the end of the 30 days, we euthanized survivors and sampled gills of the population to assess viral load and final prevalence of KHV after the outbreak, which was 100%. This is the first set of results from this challenge. The solid squares represent the mean cumulative mortality at each day with standard error of the mean. The different experimental groups are color-coded on the right. And finally, the black stars indicate statistical significance when compared to the positive control. Significance was assessed with a two-way ANOVA with repeated measures and pairwise t-test of least square means. There are 2 very exciting observations in this graph. The first is a 15% statistically significant reduction in cumulative mortality from days 25 to 30 in the acyclovir treated group when compared to the positive control. Similarly, and obviously more dramatically, the negative control cumulative mortality was statistically significantly lower when compared with the positive control from day 7 to 30. There was not a significant reduction in cumulative mortality in the cidofovir group, and it may be that the viral DNA polymerase has a narrow specificity for nucleoside analogs. And this is something that was seen before, for example, in this paper, where the same group that tested acyclovir against KHV and saw around 80% decrease in viral load, tested an acyclovir derivative and only saw a 25% decrease in viral load. With this, acyclovir does not seem like a good candidate for therapy at this time. The second observation is here in the beginning of the timeline, where we can notice that the positive control group started dying on day 1, while the treatment group's mortality was delayed until day 4. Since this graph is presenting cumulative mortality, this onset of mortality is referred to the group as a whole and does not account for first death in each pod. Because of that, we combined first death per pod for each group and ran a Kruskal-Wallis test. Each bar represents the mean time to first death in days for the color-coded group with standard error of the mean. The black stars indicate statistical significance when compared to the positive control. With this, we can see that the time to first death per antiviral treated group was not statistically significant, but I think it's definitely something to note that for 3 days, there was no mortality in the acyclovir and cidofovir groups. So keep this in mind because we will come back to it later. These are the second set of results from the challenge. The solid dots represent the mean viral load of sampled fish in log 10 of the number of KHV copies per microgram with the standard error of the mean. And the groups are once again color-coded and labeled on the x-axis. A one-way ANOVA and Tukey's multiple comparison analysis was used to determine statistical significance, which was considered at a p-value of 0.05 or less. There was no statistically significant differences in viral load among the group mortalities or the group survivals. The mortality viral load graph on the left is expected because it's logical that mortalities in the treatment group would have a viral load comparable to the positive control. In addition, the mortality viral load data confirmed KHV in every mortality from the challenge. The survivors' mortality viral load graph on the right is a little disappointing because you would expect that if acyclovir had a significant impact on cumulative mortality, that it would decrease viral load in general. One explanation is that at some point, acyclovir did have an impact on viral load, but since we were collecting at the end of the 30-day challenge experiment, the viral load of the other groups was already decreasing as part of the normal immunity response to KHV. And as you can see in this graph, this is a plausible explanation, considering a statistically significant viral load reduction when comparing survivors to mortalities by using an nested t-test. We did not have as a goal assessing the effects of acyclovir in viral load, but this data suggests that if this is a goal in a future project, sampling should be performed at various time points during the challenge. Now, for the last experiment of this project, the PK study, we used 102 koi fingerlings with 6 fish per time point, averaging 9.8 grams. Under buffered MS222 anesthesia, we administered a 10 mg per kg acyclovir dose intrasalomically. Groups were recovered and separated in tanks until their time point was concluded, and we had groups from 15 minutes to 6 days post-injection at various intervals for a total of 16 time points, plus a negative control needed for both an acyclovir standard curve and a normal histopathology analysis. When time was up, we euthanized the fish with an MS222 overdose, and blood was collected into heparin-lined capillary tubes from the caudal tail vein after severing the caudal peduncle. Then we centrifuged the tubes and collected just the plasma for liquid chromatography tandem mass spectrometry analysis. The spine was then severed just caudal to the opercula as a secondary method of euthanasia before completing a full necropsy where we collected samples of the spleen, gills, brain, liver, heart, and kidney for histopath. These are the results from the PK experiment. The solid pink dots represent the mean acyclovir plasma concentration at each time point with standard error of the mean. I hope you can appreciate that the curve indicates an acyclovir peak plasma concentration of 141 micromolar obtained in 45 minutes with a half-life of 14 hours. If we compare these values with human data, acyclovir for this same dose intravenously achieves a peak plasma concentration of 87 micromolar in about an hour with a half-life of 3.16 hours. So we can see that koi fingerlings achieve a higher peak plasma concentration in less time with a longer half-life. Because we don't have pharmacodynamic data for acyclovir against KHV, let me use the median effective concentration, or EC50, of acyclovir against human herpes viruses as a frame of reference. And it's important to mention, and those of you that work with viruses or in pharmacology may know this already, that EC50 values can be different depending on cell type used for sensitivity testing. With that said, the EC50 of acyclovir against herpes simplex virus 2 in viral cells is around 3.99 micromolar. Please note that I changed both axes to visualize the later time points better. The concentration of 3.99 micromolar is seen to be reached until about 8 hours post-infusion in humans. On the other hand, with a longer half-life in koi fingerlings, they maintain a plasma concentration above that threshold for 25.5 hours. So based on this, while humans require TID infusions, as you can see in this graph from Whitley et al., koi may need only SID injections. Now, this is where that delay in mortality may be a cool clue to consider. As we just discussed, the EC50 can vary a lot between viruses, viral strains, and cell lines. So it's possible that the EC50 of acyclovir for KHV is lower than 3.99 micromolar. In this case, redosing the 10 mg per kg acyclovir intrasalomic dose every 2 to 3 days may be sufficient. But obviously, this is only an educated speculation until we can perform a multi-dose trial or a pharmacodynamic evaluation. Additionally, histopath of spleen, gills, brain, liver, heart, and kidney of the acyclovir-treated koi detected no lesions associated with tissue toxicity when compared to untreated koi. And this suggests that acyclovir is non-toxic to tissues, at least with short-term use. So to bring all of our conclusions together, these results suggest the following. Number one, that acyclovir is non-toxic to cells and tissues at the tested doses after short-term use. Number two, that a single 10 mg per kg intrasalomic acyclovir injection is safe and effective at reducing cumulative mortality during a KHV outbreak in koi fingerlings, while a single 5 mg per kg intrasalomic dose of cidofovir is not. And number three, that acyclovir has a high peak plasma concentration in a short amount of time in koi fingerlings with a slow decrease in concentration and a long half-life. And that this may account for the delay in mortality in the beginning of the treatment. I do want to mention that age-related changes in drug kinetics have been reported in koi for other drugs. So it's something to have in mind when treating adult koi based on this data. We are getting ready to run a multi-dose protocol very soon, COVID-19 permitting, and hopefully a pharmacodynamics experiment is something to consider down the line. Finally, I want to acknowledge some institutions and people that were crucial in the success of this project: to the California Academy of Science, the Monterey Bay Aquarium, the Hub SeaWorld Research Institute, and the William R. Pritchard Veterinary Medical Teaching Hospital at the University of California, Davis for their support of the Aquatic Animal Health Fellowship that brought me here. to the Center for Aquatic Biology and Aquaculture at UC Davis, particularly to Linda and Matt for their constant support with the housing and care of the fish, to the Center for Companion Animal Health at the School of Veterinary Medicine in UC Davis for sponsorship of this project, to Heather Nitch and her lab for the plasma analysis of the PK study, and last but not least, to the entire Aquatic Animal Health Lab, particularly to Susan, Zainab, Rushu, and Esteban for being in there for the long 6 a.m. to 7 p.m. days, helping throughout all stages of the experiments in fish care. Thank you so much. These are some of my references, and thank you for tuning in. I'll take any questions.
Very interesting talk, Eva. Thank you very much for providing that. I don't see any. Feel free to unmute yourself if you've got a question, or you can put it into the chat box and I can read it to her.
Hi, this is Alex Primus.
Hi.
Eva, very nice talk. It was a very nice piece of work.
Thank you.
Just considering kind of administration routes and what might be best for fish, have you considered looking at, even just the pharmacokinetics of oral administration?
It's so interesting that you say that. We actually have samples collected from an oral gavage trial that we did at the same time we were doing the injectable one. It's just because of time constraints and the expense of a PK study with so many time points, we limited the first part of our experiment to intercelomic data. But we do have the samples collected and hope at some point to run those and have some data to share.
Great, thanks.
This is Dave Scarf. Eva, very nice presentation.
Thanks, Dave.
And there's some interesting things. My compliments, by the way, to try and pack that much information into 10 minutes. It's very confusing. I'm going to make an assumption, but I'd like to look at the data. It will take some time to look through it, that acyclovir may actually assist in decreasing mortalities and viral loads. If this is the case, considering the cost of acyclovir, how viable is it for a veterinarian to extra label and pass the cost on to a client?
Acyclovir is actually one of the cheapest nucleoside analogs available. I think I calculated 4 cents per milligram or something like that. Cidofovir, for example, it's like $4 per microgram for milligram. So the fact that acyclovir was the one that was successful is really fortunate for us as veterinarians because the cost won't be an issue.
Okay,
that's a great question.
I suspected as much. The second part of that too, how practical do you think it might be once a producer has discovered KHV in their population for veterinary intervention and the use of acyclovir? How practical is this?
Yeah, we talked about this when we were discussing the results, because I think it's sort of like an ethical debate when we talk about providing veterinary services for breeders and distributors of koi for pet purposes, because then you would be selling KHV positive fish. And so I think at this point, if we were to diagnose KHV in a breeder or distributor, the recommendation, the most responsible recommendation at this point would be to call. But if we're talking about people that have their pets in their ponds and their beloved pets, and they are informed about how these animals will remain KHV positive for the rest of their lives, it is our responsibility to do informed recommendations, and then the owner can make their own choices that are informed based on their needs.
Okay, thanks. You really answered the third part of that is, as long as people recognize that once infected with any of the herpes virus, the host will keep that virus in their neural system until they are stressed and then shed like crazy. Unfortunately, I'm one of those candidates for shingles, and I can promise you it's a very unpleasant type of thing to have. Same works with koi. Thanks very much for your presentation.
Of course, you're welcome. Thanks for your questions.
Hi, Eva. This is Tom Locke from Michigan State University.
Hey.
I'll just echo everybody else. You did an excellent job. So I'm wondering on your virus loads. I know that it's very tricky to culture cyprinid herpes virus 3, but I'm wondering if you would expect that you might have seen different results in virus loads had you, instead of using qPCR, had you used tissue culture to look at active versus inactive virus.
Yeah, I don't think we saved any tissue preserved in a way that would be good for that. We did save some plasma for virology, but we have not run those samples. So it would be interesting to see if we can culture some of those and see what kind of results we get.
Thank you.
Thank you.
So Eva, we have a question here in the chat box from John Drennan. He's wondering if you could clarify how the dose was administered and if it was IP injection, could mixing with cocoa butter extend half-life? Basically a time delay release.
Interesting. I have no idea about the cuckoo butter. I would love to read about that. We injected the dose just cranial to the pelvic fins, and it was just as a solution in PBS. So it would be interesting to see if there are compounds that can extend the half-life. Acyclovir and many of these nucleoside analogs, unfortunately, are notorious for having a short life in most species. So humans, if it wants to be effective in preventing shedding and preventing outbreaks, it has to be taken 3 times a day, every day for their lives. So this is something that we know it's a limitation with acyclovir and those types of compounds, but if it was effective, it would be very good for pet koi fish that right now don't have any treatment options in the US.
Great, well, thank you again, and virtual round of applause for you. If you could go
Thanks,
ahead and,
Nick.
yeah, no problem.